Thanks, David. I want to thank Lena for the opportunity to be here this afternoon, and certainly want to thank Smart Tots and IRS for sponsoring this panel. So today I want to talk to you about the translational approaches that are potentially out there to treat anesthesia-induced neurotoxicity. So as you've heard, the animal evidence is pretty irrefutable with regard to neurodegeneration and impairment in memory and learning. And the clinical studies have certainly demonstrated an association that is concerning. Although not definitive yet and hasn't been causative, there's certainly heightened awareness about this in the patients we take care of. So I believe that now is the time that we should really explore the potential therapeutic interventions to prevent or treat anesthesia-induced neurotoxicity and protect the developing brain. So there have been several different therapeutic approaches that have been attempted successfully in the preclinical models, and we're going to go over them, the majority of these, in the next few minutes. So first, just to talk about the mechanism, from an apoptosis standpoint, which seems to be a predominant toxic effect of anesthetics in the developing brain, the upstream mechanisms are not widely known, but the downstream mechanisms are fairly clear. It's largely mediated by activation of the intrinsic pathway of apoptosis, where there's cytochrome C release from mitochondria forming the apoptosome with APAF1, which activates caspase-9 and then activates the effective caspase-activated caspase-3. And that leads to DNA breakdown in the nucleus and then programmed cell death. To tease the pathway down a little bit more detail for the remainder of the talk, I'm just going to go through some of the intrinsic pathway here, which is on your right, which is also called the mitochondrial pathway. You can see that there are several pathways that are interconnected here which can lead into activation of the mitochondrial pathway. If you look at the top there, you see BDNF, which is a neurotrophic factor, which is brain-derived neurotrophic factor, which is a very important growth factor in the developing brain, which promotes pro-survival in neurons. Anesthetics have been shown by Vesna's group and others to impact BDNF and decrease BDNF, and that would lead to programmed cell death. It's a minor pathway, but it's certainly partially to blame as far as cause. There's also this pro-survival kinases such as AKT, ERK1/2. These feed into Bax translocation and Bax permeabilization of the outer membrane. You see in the bottom panel there, as well as in the mitochondria in the yellow. Bax permeabilization of the outer membrane of mitochondria actually permits cytochrome C to be released, which then forms the apoptosome with APAF1 and activates caspase-9 and 3. Importantly, you see in the middle there BCL2 and what's not depicted is BCL-XL. Those are two anti-apoptotic proteins which bind to inhibit Bax permeabilization, and they actually prevent release of cytochrome C. And then you see tumor suppression protein there on the left bottom of the top schematic, which also is a pro-apoptotic protein. Now, in the boxed inset there, you see upstream from Bax translocation, cytochrome C, which is in red, is bound to cardiolipin on the inner mitochondrial membrane. In the presence of reactive oxygen species and hydrogen peroxide, cytochrome C can actually peroxidize cardiolipin and can lead to cytochrome C being liberated from that complex, and then with permeabilization of the outer membrane of the mitochondria by Bax, cytochrome C can then be released. So these are the intrinsic components of this pathway, and we're going to go over the mechanisms of some of these therapeutic interventions. All right, so let's talk about preconditioning first. So what is preconditioning? It is the process by which tissue protection is conferred following a brief exposure to a mild stimulus, which then leads to temporary resistance to a subsequent, much more severe stimulus or injury. Now, preconditioning has been shown with ischemia, hypoxia, hypothermia, pharmacologic manipulation, a variety of tissues and organs, giving them protection, and this is including heart and brain. Preconditioning with volatile anesthetics has been shown to occur and has been shown to limit brain injury in models of stroke and cerebral ischemia. So investigators started to wonder, well, maybe preconditioning can protect the developing brain from anesthesia-induced neurotoxicity. Well, the mechanisms of preconditioning that have been demonstrated previously are an increase in the BCL2 Bax ratio, which would promote anti-apoptosis, downregulation of the tumor suppressor protein p53, which would also promote anti-apoptosis, generation of the good, and its paradoxical generation of the good reactive oxygen species, which are important for signaling and are probably critical to the preconditioning response. Preconditioning also induces mild endoplasmic reticulum stress and activates a number of pro-survival pathways such as the AKT pathway, adenosine A1 receptor, protein kinase C, and ATP-dependent potassium channels. So where in the pathway does preconditioning work? It's going to upregulate and increase the anti-apoptosis protein BCL2, and it's going to decrease Bax translocation to promote anti-apoptosis. Also, there's a downregulation of p53 tumor suppressor protein, and there's an upregulation of the good kinds of ROS, which promote survival and signaling. Also, there's an upregulation of the AKT pro-survival kinase pathway. So what's the evidence of this in anesthetics with anesthetic toxicity? So this was published in 2007 by Wafeng Wei at UPenn, and what they did was they looked at primary cortical neuron cultures. They exposed the cultures to 24 hours of isoflurane, and in a subgroup of the cultures, they exposed the cells to 1 hour of 2.4% isoflurane prior to that 24-hour exposure. And what they found was, if you look at the left side is MTT, they looked at the ability of the cells to reduce MTT, which is an indication of survival, and a reduction in MTT reduction is indicated of impaired cellular function and a marker of cell death. And what they found was that in the dark gray bars, that the cells that were exposed to isoflurane by themselves had reduced MTT, reducing ability, indicating that they were dying after the isoflurane exposure. And you see the hatched bars, cells that were exposed or preconditioned with 2.4% isoflurane had increased ability to reduce MTT and pro-survival. On the right-hand panel, you see LDH, and that's they measured LDH in the serum of those cells in the medium. And LDH in the medium is indicative of cellular lysis and cell death. And you can see the isoflurane-exposed cells alone had marked increase in LDH in the medium on day 7 and day 16 of in vitro, and that isoflurane preconditioning protected those cells. On the left-hand panel, you see here that they also found that the preconditioning response was a dose-dependent response, where when cells were exposed to preconditioned with 2.4% isoflurane or 1.2% isoflurane, there was protection with regard to MTT compared to the black bar, which is isoflurane alone. And the preconditioning response was lost at about 0.6% preconditioning with isoflurane. Importantly, they also found that preconditioning was not limited to just isoflurane. Cells were able to be preconditioned with halothane as well as sevoflurane. In vivo, this is work out of UCSF that was published in 2010 in anesthesiology. The group demonstrated that xenon pre-treatment attenuated the anesthetic-induced apoptosis response in the developing brain of rats. And what they did was they exposed animals, 7-day-old rats, to 2 hours of 70% xenon as a preconditioning agent, and compared that to preconditioning with nitrous oxide or hypoxia. They then exposed the animals to 70% nitrous oxide with 0.75% isoflurane for 6 hours, and they evaluated activated caspase-3. And you see in panel A there, that's a normal amount of activated caspase-3 in the cortex. Below that is and B is the animals that were exposed to anesthetic cocktail alone. There's an increased amount of caspase-3 in the neocortex. Xenon appeared to protect, xenon preconditioning appeared to protect activation of caspase-3 in the neocortex and hippocampus. And panel D and E show you nitrous oxide preconditioning and hypoxia preconditioning. And nitrous oxide had very little effect, as you see in the graph below, third from the left. And hypoxia actually may have worsened the amount of activated caspase-3 in the brain. But the xenon preconditioning actually significantly decreased the number of activated caspase-3 cells in the brain, in the cortex, as well as in the hippocampus. They then looked to see about mechanism, and they found with Western blot analysis that xenon preconditioning increased steady-state levels of that anti-apoptosis protein BCL2 in panel A. Hypoxia actually decreased steady-state levels of BCL2, which would promote apoptosis. When they looked at cytochrome C in cytosol, they found that xenon had no different levels of cytochrome C release compared to animals that were exposed to no agent. And nitrous oxide and hypoxia actually led to an increase in cytochrome C release. In the bottom panel there, P53, the tumor suppressor protein, xenon decreased steady-state levels of P53, and hypoxia actually increased them. So the decrease in P53 would promote anti-apoptosis, and the increase would promote apoptosis. So then they looked at the effect of preconditioning of xenon on memory and learning using foot shocks and fear conditioning at 40 days of age, and found that in panel A and panel C, that nitrous oxide and isoflurane by themselves reduced the amount of freezing time of these animals after training, indicating an impairment, an acquired impairment in memory and learning. But xenon actually prevented that decline in memory and learning, and hypoxic preconditioning actually worsened it. Okay, so preconditioning actually may be something that should be explored as a potential therapeutic intervention. So let's switch gears and talk about dexmedetomidine. Dex is a very commonly used agent now in pediatric anesthesia. You all know that it's a central alpha-2 adrenoreceptor agonist with a variety of positive properties such as sedative, analgesic, and sympatholytic properties. Importantly, alpha-2 adrenoreceptors play a role in activating a number of survival pathways in the developing brain. And Dex has been shown to be a neuroprotectant in a variety of insults and experimental models of stroke and cerebral ischemia. The mechanisms that have been attributed to this neuroprotection is that Dex upregulates the anti-apoptotic proteins MDM2 and BCL2, upregulates neurotrophic factor BDNF expression, increases phosphorylated Krebs, which is pro-survival, and enhances pro-survival ERK signaling. So in the pathway here, Dex will increase BCL2, the anti-apoptotic protein will enhance BDNF signaling, which is pro-survival, and will increase ERK signaling as well. And this work done in rats by Mervyn Mays' group in UCSF demonstrated that Dex was able to attenuate isoflurane-induced neurocognitive impairments in neonatal rats. What they did was they gave 3 doses of intraperitoneal dexmedetomidine in animals at 7 days of life. These were rats that were exposed to 6 hours of 0.75% isoflurane. And they found that Dex limited the isoflurane-induced caspase activation in a dose-dependent manner in the hippocampus and thalamus as well as the cortex. And what you see is in the panel A, you see a normal healthy control that received saline. This is the normal amounts of activated caspase 3 in the neocortex. You can see some staining there, which are the black cells. And then isoflurane exposure caused a significant increase in activated caspase 3 expression in the panel B. And then the animals that got 1 mic per kilo of dexmedetomidine, 3 doses, you can see that it attenuated the activation of caspase 3 in the neocortex in panel C. And then in the graphical quantification, you can see that different concentrations of dexmedetomidine significantly decreased caspase 3 activation in both the cortex and hippocampus in a dose-dependent fashion, going from 1 mic to 10 mics to 25 mics. And then when they gave APT, which is an antagonist of dexmedetomidine, they could partially abrogate that effect. They then looked at the effect of dexmedetomidine in this context on memory and learning, again with foot shocks. And on day 1 in panel A, you see they found no difference in short-term memory. But on day 2 of testing, they found that the animals that had isoflurane alone with saline control had an acquired impairment in freezing time with foot shock, but that dexmedetomidine with isoflurane actually prevented that acquired defect. So dexmedetomidine may be protective. Let's shift gears and talk about glycogen synthase kinase 3 beta. And this is a very important protein which is known to be important for memory formation. When it's inhibited by phosphorylation, it actually promotes long-term memory. and this is via axonal growth and neurotransmitter release, and low activity promotes memory formation. However, when it is dephosphorylated or unphosphorylated, it is constitutively active and has high activity and actually can impair memory via impairing axonal growth and impairing neurotransmitter release. This is a really nice study that came out in 2010 by Liu and colleagues, and what they found was that when they inhibitory avoidance training with foot shocks in rats, that over time there was an increased amount of GSK3 beta phosphorylation consistent with memory consolidation, and you see that in the Western blot there. From 30 minutes to 2 hours. The maximal phosphorylation was seen at 2 hours after training. Well, importantly, when they exposed animals to 2% sevoflurane after training, sevoflurane actually prevented the phosphorylation of GSK3 beta, which suggests that this is a mechanism of impaired memory formation. Sevoflurane by itself, without training, had no effect on the phosphorylation state of GSK3 beta, you can see in the Western blot, but it is the prevention of the phosphorylation which may be very important. So the investigators considered lithium, and lithium is a very interesting drug. It is a well-known inhibitor of GSK3 beta. It has been shown to protect against drug-induced and hypoxia and ischemia-induced neuronal apoptosis in the developing brain. So the investigators took the animals and they pre-treated them with lithium, and then did the inhibitory avoidance training with foot shocks and exposed animals to 2% isoflurane, and then they measured the amount of GSK3 beta phosphorylation. And what they found was, in the context of training and sevoflurane, that lithium actually preserved the phosphorylation state and the inhibition of GSK3 beta, promoting memory consolidation and preventing the inhibitory, impairment, the acquired memory impairment. And sevoflurane alone there, you see, actually again prevented phosphorylation of GSK3 beta. And this is the effect on memory retention. You see that the animals that got sevoflurane with lithium had preserved memory retention latency, where the animals that got sevoflurane alone had acquired defects in memory retention. So lithium may be a drug that should be explored further, since it is given to a variety of different patient populations. All right, let's shift gears and talk about vitamins and dietary supplements. Oxidative stress is a known effect of anesthetics. Virtually all anesthetic agents induce inflammation and oxidative stress in a variety of tissues, including the brain. They also increase expression of a variety of pro-inflammatory cytokines, such as TNF-alpha, IL-1 beta, IL-6, and they induce a variety of different oxidants, such as reactive oxygen species, reactive nitrogen species, and the reactive oxygen species hydrogen peroxide. And not surprisingly, a number of vitamins and dietary supplements possess known anti-inflammatory and antioxidant properties, and so investigators have started to look at the effects of some of these common nutrients on anesthesia-induced neurotoxicity. Vitamin D3 is of interest because it actually induces calcium binding protein expression and stabilizes intracellular calcium in cells. And it has been shown that NMDA antagonists, for example, do not activate caspase-3 in cells that express calcium binding proteins. So one would hypothesize that vitamin D3 may have a protective effect. And in fact, that's what Turner and colleagues demonstrated in 2012, is that vitamin D3 pretreatment in 7-day-old rats, 24 hours before 20 milligrams per kilo of ketamine, actually limited the activation of caspase-3 in the developing brain in the somatosensory neocortex. You see the light gray bar there is the animals that got ketamine alone, and the darker gray bar are the animals that got vitamin D prior to ketamine. You see on the left-hand side of the graph there that vitamin D by itself had no effect on apoptosis, or at least with caspase activation. Let's switch gears and talk about melatonin. This is work out of Vesna's lab. Melatonin is a hormone that regulates circadian rhythm. It's secreted by the pineal gland, important for regulating the body's sleep-wake cycle, and it is known to have antioxidant properties. So what this group did was they assessed the treatment with melatonin in P7 rats, getting a cocktail of midazolam, nitrous oxide, and isoflurane. And what they found was a dose-dependent inhibition of caspase-3 activation in the developing brain, in the anterior thalamus, and in the cortex of animals getting the anesthetic cocktail, with the majority of the effect at the higher end of the dosing at 10 and 20 milligrams per kilo subcutaneous. So melatonin may have some effect. Mechanistically, how does melatonin work? What they found was that it limits cytochrome C release there in A and B in the thalamus and the cortex during a 4-hour and a 6-hour exposure. And it also upregulates the anti-apoptosis protein BCXL, you see on the right side of the panel there in A and B, A and C, at also between 2 and 6 hours of the anesthetic exposure. So in the pathway, melatonin seems to reduce cytochrome C release and upregulates the anti-apoptosis protein BCL-XL. L-carnitine is another agent that has been assessed. L-carnitine is a lysine derivative, which is important for long-chain fatty acid transport into mitochondria. Fatty acids are important for ATP synthesis via beta oxidation, so they're very important for bioenergetic capacity of cells. L-carnitine has been shown to attenuate neurologic injury acquired with inborn errors of metabolism as well as inherited mitochondrial cytopathies. And it has a protective effect due to reducing the decay in mitochondrial function, as well as preserving the mitochondrial membrane potential, cardiolipin content, and the bioenergetic capacity, ATP synthesizing capacity of mitochondria. Well, L-carnitine was given in various doses to rats during a 6-hour anesthetic exposure with nitrous oxide and isoflurane, and you can see that L-carnitine had a significant effect on activation of caspase-3 in the developing brain, but mostly at the higher doses, at the 300 and 500 milligram dose. The smaller doses had little or no effect, and L-carnitine by itself seemed to have no effect on caspase-3 activation. So another agent that has been explored. Mechanistically, they assessed for BCL-XL and BAX expression with Western blot and found that nitrous oxide with isoflurane by itself reduced the BCL-XL protein expression and decreased the ratio, whereas carnitine actually preserved the ratio, so anti-apoptosis. So it promoted the anti-apoptosis ratio of those proteins. Omega-3 polyunsaturated fatty acids, a group of essential nutrients that are known to be important for normal brain development. Prenatal supplementation of omega-3 fatty acids have been shown to provide neuroprotection in newborn rats exposed to ischemia and hypoxia as well as hyperoxia. And what Lay and colleagues did in 2013 was to administer it to pregnant dams up until 2 weeks postnatally. And what they found was that it prevented caspase-3 activation in the brain of animals exposed to sevoflurane. And then when they looked at memory and learning, they found beneficial effects with regard to foot shocks and latency with escape from the water maze. So omega-3s may have a positive effect. The mechanism is purportedly to be through upregulation of BDNF growth factors as well as reduction in the bad type of reactive oxygen species. All right, let's talk quickly about antioxidant therapy. This is again out of Vesna's lab with UC-134 and PPX, two very well-known antioxidants. They demonstrated that UC-134 was able to decrease DCF fluorescence, which is a marker for ROS activation in the context of an anesthetic cocktail at 7 days of life. They found that that UC was able to reduce lipid peroxidation in the animals that got anesthetic cocktail as well compared to anesthetics alone. And this led to a prevention in abnormal mitochondrial morphology. When they looked at the effect of UC and PPX on neuronal loss, they found that both antioxidants prevented loss of neurons in animals exposed to the anesthetic cocktail versus animals that received anesthetics alone. And this translated to a preservation of cognitive function. All right, the last two things I want to talk about are two very clinically translatable approaches which could be translated into practice today if neurotoxicity is found to occur in humans. The first is hypothermia. Hypothermia is a known protectant to be able to protect the developing brain from hypoxia and ischemic injury, very commonly applied to our cardiac patients, for example. It limits neuronal cell death even when applied after the insult, and it prevents early neuronal death via disruption of the exocytotoxic response. And what John Onley's group demonstrated was that reducing the body temperature in 4-day-old mice by 5 degrees from 34.7 to 29.7 actually significantly prevented activated caspase-3 in the brains of animals receiving 0.75 of isoflurane for 4 hours or ketamine 40 milligrams per kilo. And this is a profound protective response. Importantly, though, hypothermia also decreased the amount of natural or physiologic apoptosis in animals that receive no anesthetic. So there could be a deleterious side of hypothermia in this context as well, and this needs to be teased out further before we start employing in our patient population. The last agent I want to talk to you about is carbon monoxide. This is work out of our lab, and I believe IARS distributed a nice pamphlet with our paper with a nice editorial from Vesna Jeftovic-Todorovic. So carbon monoxide in toxic levels are known to cause tissue hypoxia and cellular hypoxia and can lead to death. But in low levels, it's actually quite protective and has anti-apoptotic properties and has been demonstrated in a number of different tissues and animal models. We all produce endogenous carbon monoxide in our liver via breakdown of endogenous heme via heme oxygenase. And bilirubin is the end product, but carbon monoxide is a byproduct, and we excrete this toxin in our exhaled breath. Well, importantly, in the anesthesia breathing machine, carbon monoxide is actually generated exogenously, and the offending agent in the anesthesia breathing machine is the CO2 absorbent, conventional soda lime, which can break down anesthetics such as desflurane, as shown here, producing low levels of carbon monoxide even in hydrated soda lime. And so in the anesthesia breathing circuit, where we permit or can permit patients to rebreathe their own exhaled breath during low flow anesthesia, for example, patients will rebreathe everything that they exhale except for carbon dioxide because it's actively scrubbed. So exhaled carbon monoxide is rebreathed, and we demonstrated in 2 studies that when minute ventilation is exceeding fresh gas flow, that carbon monoxide is detectable and leads to a rise in carboxyhemoglobin, so you get a subclinical exposure. Well, where does carbon monoxide work with regard to apoptosis? It actually binds to the cardiolipin cytochrome C complex, and it inhibits the peroxidation of, this is not working now. There we go. It actually inhibits that cytochrome peroxidation of cardiolipin, preventing cytochrome C mobilization and cytochrome C release. So we tested this in 7-day-old mice in the context of an isoflurane exposure for 1 hour, 2%, and we found that compared to the air-exposed and isoflurane-exposed animals in the top panels there, in the neocortex, hippocampus, and hypothalamus-thalamus, that activated caspase-3, as shown by the arrows, were significantly reduced in animals that got 5 parts per million and 100 parts per million carbon monoxide with isoflurane. And graphically, you see there's a dose-response phenomenon here with regard to carbon monoxide exposure in the black-hatched and black-speckled and the gray-speckled bars compared to isoflurane with air alone. When you looked at tunnel-positive nuclei, which is a marker for apoptosis, we found that, again, carbon monoxide, 5 parts per million and 100 parts per million, significantly decreased the number of apoptotic nuclei in the neocortex, hippocampus, and hypothalamus compared to isoflurane with air, again in a dose-dependent fashion. Mechanistically, we found that, if you look at the left panel, that carbon monoxide 5 and 100 parts per million significantly inhibit cytochrome C peroxidase activity in a stepwise fashion, dose-dependently, with and without isoflurane. And this led to, in the two right panels, a reduction in cytochrome C release into cytosol, more cytochrome C in the mitochondria of the cytochrome C exposed animals. So carbon monoxide actually prevents isoflurane-induced apoptosis. Well, what's the effect on memory and learning? We found that animals that received 5 parts per million isoflurane with, 5 parts per million of carbon monoxide with 2% isoflurane for 1 hour had preserved latency in memory and learning on day 1 of testing in a T-maze versus animals that got isoflurane alone, and this was tested 6 weeks after exposure. So it seems to preserve memory consolidation and prevent the acquired impairment in memory retention. And again, with days to criterion, the isoflurane carbon monoxide group had markedly improved days to criterion, so preserved memory. So we think that inspired carbon monoxide will limit and prevent isoflurane-induced apoptosis in the developing brain and will prevent the acquired defects in memory and learning. And so, taken full circle, that low-flow anesthesia may, targeting specific carbon monoxide levels, may actually prevent and inhibit anesthesia-induced neurotoxicity. And we're going to explore this further. So in summary, we have a number of therapeutic options that have already been tested in animals with success that we can explore further for potential application in our patients in the immediate short term if neurotoxicity is shown to occur in the patients we take care of. But future work, and this is more long term, will have to focus on novel agents and new approaches, and importantly, we may need to develop new classes of anesthetic agents that actually are non-triggering with regard to neurodegeneration and neuroapoptosis. Thank you very much for your attention.